Coming in to see you and have a basketball game tomorrow. All right, so this is a quick little video and lecture on incomplete and codominance. So we'll take a look at the same sheet. Again, I've altered the sheet, the worksheet on Mendelian genetics, the bikini bottom genetics sheet. And then what I did here in the content library, so everybody knows, is I added a little helper piece picture to talk about incomplete and codominant. So they kind of work the same way. They still have alleles, right? We do see some alleles here on each of these, which will switch colors. So we see like this red flower, right, which is its phenotype. It is red. That's its pheno, right? And this is its genotype here, 2 capital R's. So that means that both of the alleles are dominant, right? Both dominant. And then over here, we have this red flower is combined with this guy over here, which its genotype is 2 uppercase W's. So you're seeing kind of like, it's not codominance, but sometimes there's different ways to write this. And for our purposes, I'll tell you whether or not it's going to be an incomplete question or a codominant question. Oh, go ahead. Mr. Amicus is going to make a brief interruption. Do you need ring stands? Check over in that far closet. Yep. Probably all the way to the right, either in the top or bottom. There's a drawer of ring stands and clamps. Yeah, inside the cabinet. Yeah. There should be a drawer with ring stands and clamps, maybe on the bottom level. You can take them off. Yeah, and they just unscrew from the bases. Look further down though. There might be in the lower one, yeah. All right, so this guy's genotype is WW. And we obviously have a phenotype, and it is white. Okay. So the Punnett Square is already done for you for this flower combination. So we'll say that we have 2 parents, and they're both homozygous. Okay, so both are actually homozygous. So both are homozygous. Okay, and we'll say that this particular gene for color, which happens, and which all of you should actually see if you do have flowers at your home, or like annuals that you plant, right? If you do like little buckets or plants, or you have them in your garden, sometimes you'll see these variations. Especially, it's kind of interesting. You walk down the aisle, whether it's like at Home Depot or Lowe's or English Gardens or something, whatever flower place you're going to, or flower stand that you see on the side of the road, you'll see these variations of flower color for different types of plants. So we'll talk about, like, these look similar to kind of like a petunia or something, or they could be true for like impatiens, which a lot of people plant. They're actually fast growing. They get very lush and large, and they can form almost like a small little hedge too, right, in the summer. But we'll see some variation in the color. And sometimes when it's an incomplete dominance, meaning that if either of these alleles combine, they can do one of 2 things. In this case, the experience that we've given here, or the sample that we've given here, is that in the first case, when it's incomplete, what essentially that means is that we're going to get a mix, or like almost like a true mix, or blend, or more so a blend of color. don't spell it like an American, so I don't screw anybody up. There we go. Okay, I think I got it right. All right, so in this case, in the incomplete dominance, we see a blending, right? So what is the resulting? Let's first talk about what is the resulting genotype. Okay, so our resulting genotype is the R-W combo. Okay, and then what's the pheno? Yeah, so the pheno here is pink. Very interesting, right? We get this new kind of what we call hybrid, right? And the nice thing about flowers and plants is that you can get all kinds of blending. So Mendel also looked at this with his pea plants, right? He actually started seeing combinations of colors. And what did that do? Well, it obviously gives them different colors of flowers, but it also gave some slightly different features to the actual legume that you're trying to get, like the peas, right? The pods. So the pods ended up taking on different shapes. The peas within inside were some smooth, some were round, some were more oblong, some were wrinkled, right? Sometimes the pods are wrinkled, sometimes the pods were smooth. And as you know, if you ever grow any kind of peas in your garden, depending on what they look like and the size that they get, depends on what they taste like, right? So you can get sometimes with incomplete dominance, if you blend the right plants together, you can get a slightly different variation on what the plant is going to taste like. Now, this is something, even though we don't get into evolution in here, this is essentially what we call, this is evolution, but what is it? What kind of evolution? What kind of evolution are we getting here? Is it natural selection? No. Yeah, if we just let the plants do their thing, yes. But what is it? Is it natural or is it artificial? Artificial. Yeah, it's definitely artificial, right? We're selecting that these plants are going to breed together, and that's why, especially flower producers, right, like farms or places that produce, like greenhouses that produce flowers for sale, will actually hybrid them together so that people can get variations of colors when they go to buy them, right? So this is incomplete dominance over here on this side. Now let's talk about codominance. I've included kind of two things that happen here. Blood typing is a great one, right? Because what can happen is that essentially we have a couple of things that happen in blood, right? And this is something we'll probably do a little blood typing lab. Yeah. Pretty easy. We actually do it in forensics. So if you end up taking forensics in the future, we'll do another blood typing, but it's more related to a case. What I'm actually have you do is test how to know whether your blood is A, B, AB positive, right? Yeah, or O. Yeah, and you can tell. Now, the next factor is whether you're a negative RF factor or a positive, right? That's a little bit different. The blood typing kit that I have is looking at just whether it is going to be A, B, AB, or O, right? So in this case, sometimes blood types, depending on what the parents give to the child, will depend on what happens here. And so this is, in this case, what we call codominance. So in this case, codominance, both genotypes express. You can copy this over from the content when I'm done, or you can do it. Your choice. Okay. So in this case, I've given you two examples. We'll say that parent A, E1, is A blood type. Parent 2 is B blood type. They have a child, and the child comes out with, right? So the F1 for these two parents could end up being AB. Okay? That means that both proteins express. Right? That means that they will express and they will become almost like a dual phenotype, right? So example here with our flower. This is our flower example. Okay. So in flowers, sometimes we can have either incomplete dominance, or what we see here is codominance. And you may have seen flowers like this at wherever you buy flowers, right? A blending, like you'll actually see red and white together. Sometimes you'll see other colors. Now, it may depend on what type of protein that makes the color. So a lot of people ask, well, why is a color a certain color? Or flowers, right? We know why leaves and stems are green, right? Everybody know why? Chlorophyll, right? In flowers, it has to do with the proteins that get released, right? It's kind of like similar to eggs, right? When hens lay eggs, they actually lay all different types of eggs. And depending on the kind of chicken they are, will depend on what the protein that coats the egg with. So whether an egg is white, brown, some variation of white or brown, or green, or blue, or something in between, it's really just about the coating protein that she expels when she expels the eggs. Right, so that's all that is. That's why eggs have different colors. The difference between colors when they sell them in the store and you go to buy them, people sometimes want to buy brown eggs because they must be healthier, they're more natural. Not really. What makes eggs healthy is what the chickens are actually eating. Has nothing to do with the color of the egg, right? And if you actually buy eggs that are quite healthy, like cage-free, free range chicken eggs, and if you do your best to buy the cheaper brand, well, you'll actually notice when you pop the top open, they might be brown, but they're variations of brown, right? And then if you go to something more expensive, the only difference in the reason it's more expensive, people go, well, it must be better. Not really. It's they needed more money because they had to sort the eggs better. So they have people who either hand sort or there's a machine that hand sorts the color to make it look more pleasing to the consumer. So if you go buy eggs, say for instance, from a farmer's market and you open the carton, you might get like 4 different colors. White, blue, green, brown, different variations of brown, and everything in between. Okay? All right. Any questions on just the generics of codominance or incomplete? Okay. Now, how will you know if this is an incomplete or codominant situation if I give you a problem? It's because I will tell you. This is an incomplete dominant problem, okay? Or this is a codominance issue, okay? Yeah. Correct. Okay, so this is something Mendel thought very interesting, is that some colors can become codominant or incomplete dominant, others cannot. Correct. Yeah, so that's a good observation. So what would happen when they combine, right? In most cases, not all colors can be codominant or incomplete dominant. Red and white just seems to be a unique one in nature that can combine like this. Sometimes purple can do this. Sometimes if we mix like a hybrid color like a purple with a white, you can get a blending or a mix, like a codominant or incomplete dominant. In other cases, some flowers just don't because one actually is the more dominant in that case, right? They might sometimes both be codominant and complete, but this is where we get into, this is some of the tricky things that Mendel found out was not everything is so cut and dry when it comes to alleles when they combine together. it sometimes has to do with how they combine, right? That for some reason they are able to do some kind of blending of dominance. Okay, that's a good question. Yeah, it really has nothing to do with art. More so, it has to do with the proteins and the amino acids and how they combine in nature at the molecular level. Okay. All right. Let's do a problem. We'll take a look at the first one, and then we'll stop our recording. And then if you can work on a couple of problems on your own, and then we'll come back and visit each other. All right. So SpongeBob loves growing flowers for his pal Sandy. Her favorite flowers are pumpkins, are found in red, blue, and purple. Use the information provided and your knowledge of the incomplete dominance. So again, we are working on an incomplete dominance problem here. So R represents red. So if we have a red, okay, we'll say a red flower looks like this. Okay. Actually, I'm gonna do this in color because that may make it easier for us to see as we go. Okay. So red. We have blue, right? And then we're going to have purple. But again, we don't change the genotypes, do we? What are we doing with them for our purple guy? Huh? We're going to combine them together. So we're going to have one like this, and then I need to put what else with it? Capital B, right? So in this case, it's not going to be codominance. We're not going to see red and blue together. We're going to see them blend to make a new color. And in this case, we're going to get purple, a purple color, okay? So that's RB equals purple, all right? Okay, so let's take a look at the first problem together, and then we'll answer the questions that pertain to this problem. Okay, what would happen if SpongeBob crossed a pumpkin with red flowers? Let's grab our red again. Okay. Neither, because this is going to be incomplete. Yep. And then this is why we made a key. That's a good question. This is why we made a key at the top. So we don't really we're not trying to forget what we covered in normal Mendelian genetics. We're just trying to shift to a new topic within it. Does that make sense? I don't want you to forget what you learned, especially with the terminology. But now we're just kind of adding another little couple layers to it here, okay? So that's why I'll try to go slow. All right, so what else do we have? What other color pumpkin do we have? Okay, so we're going to combine with the blue. All right, here we go. And we put our blue parent over here. And now, excuse me, it asks us to complete the Punnett Square to determine the chances of each flower. All right, so first things first, let's play out our red alleles, since we wrote them first up here with the purple flower, right? And we can kind of already see what's going to happen. So we're going to place our alleles in the boxes, right? And we'll grab our blue allele again here, and we'll pull it over. And then we're going to grab our second blue allele, pull it over into the boxes. Now, in this case, we've already been told that this is a incomplete dominance problem, all right? So, given the genotypes and phenotypes, give the genotypes and phenotypes of the offspring. All right, so here's where I'll just switch colors here. I'm going to switch actually to this one. We know that it is going to be RB is our genotype, and our phenotype is going to be purple. Very good. How do we know this? Well, don't forget we made a key up here at the top. All right, so we've kind of set ourselves up for success. All right, so now let's answer our traditional questions that we've been answering all along about each of these. How many of the plants would have red flowers? 0%. How many of the plants would have purple flowers? 100. And how many have blue? 0. Okay? Now, I guess what we should do is maybe talk about what happens. Let's do one more problem, and let's talk about what happens when this F1 generation right here, right? This is our, right, F1 generation, these 4 offspring. What happens when they combine together? Okay? All right. So, what would happen if SpongeBob crossed 2 pumpkins with purple flowers? So we're going to switch back to our normal colors here. We'll put in our red alleles first, right? So we'll say R and R and B and B, okay? And what we'll do is we will combine together our thing. So again, this is, I'm going to go back to my black here just to signify, I kind of wrote that oddly, different than before. This is P1, right? This is our P1. This is P2, but they're both from the F1 generation up here, right? Okay, these incomplete dominant hybrids, right? These are both what? What kind of parents are both of these? Color. They're incomplete dominants, but what color? What's their phenotype? They're both purple. Very good. Yeah, not the alleles, just the whole thing when they combine together, right? So in this case, in an incomplete world, right, we're going to say that both of these are purple. All right, so now here's what we do is we're going to go ahead and combine our alleles back together. In this first box, R going to get pulled down, R pulled across. Oh, interesting. So we're going to list our genotypes over here. So we already have an R and an R. Okay. Next one, we're going to pull this R down here. And we're going to pull over the B. Like that. So we have a new allele pairing, R and B. Okay. Actually, we'll change that up to this. There we go. Okay, we're going to do the same thing. We're going to go combine these 2 alleles. We're going to take R over here, combine, and we'll pull B down. Okay, and then same thing over here. Now we're going to take the 2 B's, pull them down. Oh, interesting. So now we have, actually, what do we see now for the, so these are our genotypes. What's our phenotypes here? We got red, purple, blue. Okay. And now we can answer our percentage questions again. Okay. How many plants have red? 25%. Good. Purple? 2%. And how many have blue? 25%. Very good. All right. All right. So at this point, we will stop there for demonstration. And then what I'm going to have you do is work on questions 4, 5. Let's try to do let's do 4. We'll see how everybody does. And then we'll kind of tackle another one here. We got some slightly different alleles happening in the next problem set. In this case, for incomplete dominance. And we'll have some different ones to work on after number 4.